Good morning. I'm Christine Seidman. I'm a professor of medicine and genetics at Brigham and Women's Hospital and Harvard Medical School, where I lead a cardiovascular genetics center taking care of patients with advanced genetic disorders and also a research laboratory. In this session, we're going to talk a lot about the big elephant in the room that has yet to be completely conquered, namely, how do we ensure that these wonderfully productive gene and cell therapies are also safe? And to do that, I'm going to ask our panelists, who will introduce themselves as we go through some questions, some of the issues that they've been considering in these approaches. But I hope that we'll be able to engage you as you think about questions. So please line up those iPads and get them ready, because I'm ready to receive them. We all recognize that some of the preclinical models aren't necessarily the best way to address what will happen when we deliver therapies to patients. So we'd like to hear about what their approaches are to thinking about the right preclinical models. Additionally, although these therapies may be acutely helpful, they may need to be redosed, and we're going to hear a little bit about their thoughts on this issue. I also would raise the possibility that we should consider whether these therapies will ultimately result in long durability and even cures. That, of course, then raises the issue of whether there are going to be long-term side effects. And we'd like to hear whether people think those are theoretical or something that needs to be actively considered and long-term and require long-term modeling. So without more ado, I'm going to turn over these discussions first by letting each person introduce themselves, and then we'll come back with some really interesting questions. So to my left is Rick Fair. Rick, please help us out.
Thanks, Cricket. Hi, everyone. Good morning. Thanks for having me here. I'm the CEO of Bellicum. We're a platform company using molecular switches and novel signaling molecules to try and enhance the performance of cell therapies. We've currently applied our platform to autologous CAR T cells in solid tumors. Nice to be here.
Zandy.
Good morning. I'm Zandy Forbes, and I'm the CEO of a gene therapy company called Meira. We have a clinical pipeline with a pivotal program partnered with Janssen in a rare eye disease, but underlying our clinical pipeline are broad capabilities in genetic medicines, ranging from looking at the viral vector design, from promoters, to being able to actually control when that vector is expressed or not, as well as manufacturing, where we now have in-house GMP manufacturing, ranging from upstream, we produce our own GMP plasmid, all the way down to the analytics for release and stability, which by the end of this year we'll have brought in-house.
Sekar.
My name is Sekar Kathiresan. I'm co-founder and CEO of Verve Therapeutics. We're developing a pipeline of gene editing medicines to treat cardiovascular disease.
And Rick Modi.
Thanks, Cricket, and thanks to the organizers for having me. I'm Rick Modi. I'm the co-founder and CEO for Affinia Therapeutics. We're a next-generation gene therapy company. Much of our focus is on new capsids, new promoters, and new manufacturing technologies to get better tissue tropism and cell specificity as well.
Great. So Rick Fair, as we begin to think about really addressing the considerable burden of solid tumors, the off-target effects, the potential for high dosing needed to get efficacy and penetration of these large tumors can be really complicated. So we'd like to hear about what you think about in the context of safety and what you're doing to address these issues.
Sure. So in the oncology setting in cell therapies, as most of you probably know, the things you're worried about are immune activation syndromes like CRS or ICANS, or these on-target off-tumor effects, which are particularly acute, as Cricket points out, in solid tumors where we don't have a lot of tumor-specific antigens. And as we create more potent constructs to try and achieve greater levels of efficacy, these safety issues could become more profound. So what can we do? What are we doing in the space, and what are we doing at Bellicum? First, better treatments. So tocilizumab and corticosteroids have been really useful tools helping to mitigate these immune toxicities. They've become standard of care. I think we heard earlier from Marcella Mas about some encouraging early data about using these prophylactically. And we've also heard interesting anecdotes about using other treatments, targeting other cytokines or tyrosine kinases to potentially develop better combinations or treatment algorithms that will help us treat these immune adverse events should they occur. I think the other categories of things that we're doing are really about engineering or selecting better cells. So, starting with better targeting, moving from single antigen targeting to either dual targeting or logic-gated targeting so that we can develop cells that are more specific to tumor and therefore less likely to create systemic activation issues or on-target off-tumor effects. There's some encouraging early work there. I think the second category of improvements in the cells themselves are really about finding better cells or perhaps safer cells. We've started and seen great benefits so far using T cells, but we've seen these immune toxicities. There's lots of encouraging work going on in NK cells, in NKT cells, and selecting certain subpopulations like gamma delta T cells that may lead to highly effective effector cells, but potentially have better safety profiles. And I think the last category is really about trying to achieve better control of these therapies by using genetic modifications that allow us to manage the cells in vivo to try and both enhance efficacy and mitigate toxicity. This is obviously something we're doing at Bellicum with our molecular switch technology. We have 2 clinical programs today in solid tumors with CAR T cells where we have embedded molecular switches that allow us to repeatedly activate co-stimulatory molecules in these cells, hopefully leading to better efficacy and resistance to T cell exhaustion within the solid tumor microenvironment. But should these cells start to create adverse events, we can withhold the activating small molecule to allow the cells to become more quiescent. And should there be a high-grade adverse event, we've incorporated our apoptosis safety switch, administering a second small molecule to eliminate these cells altogether and really stop the adverse event in its tracks. So I think these are sort of the broad categories that the field are using. I'm optimistic, Cricket. I think we're making great progress, and given all the things that these great novel concepts that are in motion, I think we're going to get to both more effective and safer therapies.
Great, great. Sandy, as we think about using viral vectors, there are lots of considerations that rise to the surface, especially when the FDA is looking into the data. And I'd be curious how you're addressing these issues of toxicity, both acute and potentially long-term.
Thank you, Cricket. If we just step back for a minute and think about the theoretical safety of in vivo gene therapy, which is if you're able to deliver a copy of the gene into a cell of choice and express it in the right place, that should be fundamentally safer than many other types of drugs. And there is a concern for safety today that when you give large doses, or you may start seeing immune responses to certain doses of vectors, and there is this discussion with the regulatory agencies around some of these side effects or safety issues that are being seen. But if you really look at those, the current issues which are emerging tend to be related to the dose. So the dose is pushing up against a dose of virus that alerts the body to its presence, and that has a negative response. So one of the first things we do in addressing that safety issue overall is make more potent, much more potent viral vectors. And we take many approaches to this, but we look at the promoters, the enhancers, we look at the codon sequence, and we have many platforms for designing and developing and discovering from the entire genomic data, promoters and enhancers and regulatory elements that will drive appropriate levels of expression, which may be high for a very potent vector, and in the right cells at the right place. And that we put into all our vectors. In addition to that, the capsid, which can be used to deliver to certain tissues rather than other tissues. We have capsid screens in NHPs. We don't really treat this as a tropism method, but really to enhance transduction in certain tissues over others. We achieve cell specificity through the promoters. So we're able to, just from what I've just said, those platforms that we have, to improve potency, not by 20% or 50%, by up to 3 logs. And this is consistent over many vectors that we've looked at and optimized. So first step in making something safer is make it active at a 2 or 3 log lower dose. And in addition to that, all of that control, we, like Bellicum, have now developed a method of very, very tightly controlling how much gene product each of these vectors makes with a synthetic mammalian riboswitch, which works at thousands of fold dynamic range. And with oral small molecules, we can precisely determine exactly how much of that product is produced in every single organism. Mice at the moment, but eventually people. So we look at a number of different things in the vectorology. And of course, I mentioned we bought manufacturing in-house.
Yeah.
Clearly, as we've developed our manufacturing processes that are applicable to all of the vectors that we make, we are able to produce safer, more consistent batches. We're currently in a pivotal, so we've discussed our manufacturing processes with agencies around the world, and we've learned a huge amount about what they want to see that allows them to understand that every batch of your product is safe and effective, as you've shown it to be in the clinical studies, and you can show that with your QC. So we look at everything we can, because safety is clearly important in the developing of any drug, and particularly important when you're addressing regulatory agencies.
True, very, very, very true.
Yeah.
Sek, when we think of other types of vectors, nanoparticles and the like, I'd like to hear your opinion about the safety issues of those in comparison to viral vectors, but also a little bit more about the cargo. When you are perturbing a somatic cell's DNA, what's the likelihood and chances for having a bystander, a nearby effect, or a completely off-target effect? And how do we address that long term?
Yeah, that's a great question. So I think take a step back. Verve is focused on atherosclerotic cardiovascular disease, so heart attack, the leading cause of death in the world. So we're trying to apply gene editing to this very common problem. And what's the unmet need here? Well, it's been known for probably about 20, 30 years that the main way to treat, and also the main way to prevent a first heart attack, is to get somebody's LDL cholesterol, the so-called bad cholesterol, as low as possible for as long as possible. Right now, we're trying to do that with daily pills or intermittent injections, and that chronic care model is broken. Less than half the patients actually are on statin medications to lower LDL cholesterol, even after having had a heart attack. And most people's LDL is not well controlled during using the chronic care model. So the thesis behind Verve is that could you actually change all that by having a one-time treatment, permanent lowering of LDL cholesterol that's efficacious and safe? And where did this idea come from? Well, this idea comes from human genetics of the type that Cricket and I used to do, which is that human genetics has shown that if one's LDL is really low lifelong, you're not going to get a heart attack. And some people are walking around who naturally have their LDL very low, and they have it so low because they have a gene turned off naturally, a cholesterol-raising gene turned off naturally in their liver. So our gene editing medicines are basically looking to mimic that natural situation, that natural resistance mutation. And so the first aspect of safety for our product comes from the fact there are humans who naturally have our target, PCSK9 in the first case, turned off, and those humans are healthy and they're resistant to heart attack. So the target safety is probably the first step. So The second question is, okay, how are we going to actually mimic that natural situation? So the idea we came up with back in 2018 was to edit the liver DNA to basically make a single spelling change that would lead to turning off the cholesterol-raising gene. And that's what our first product does. It's a gene editor, specifically a base editor, that essentially goes to the PCSK9 gene sequence, makes a single spelling change, one base pair change, to turn off the gene. And when we turn off the gene, the blood protein level comes down by about 90% at 2 weeks. The blood LDL comes down by about 70% at 2 weeks. And then 2 years later, the LDL is still down 70%. So it works right now in non-human primates. And in Cricket's question about what about the safety, both from the acute infusion, this is a one-time intravenous infusion that gets the editor to the liver, what's the safety of that, and then the long-term safety, specifically around on-target versus off-target edits. And so what we are seeing right now with the preclinical data in human cells, in mice and monkeys, is that the acute infusion is well tolerated. It's an acute infusion of the mRNA for the editor, the guide RNA, both of those packaged in a lipid nanoparticle. That's our drug product. It's well tolerated. And then in primary human cells, the edit is very precise. There's a single base pair change in the PCSK9 gene to turn off the gene, and then really no observed editing anywhere else in the genome that we've looked at. So right now, that's the data, and we're on track to take this product to patients in the second half of the year, our first in human study in patients with a genetic form of high LDL and premature heart attack called heterozygous familial hypercholesterolemia. And those will be the first patients treated. And we are, of course, looking forward to translating the data from non-human primates to patients this year.
Great. Rick, Modi, way down here at the end. I'd be curious to hear about how much we can really manipulate the capsid proteins of viral vectors and how that can improve safety. And in particular, is this going to be an opportunity to do redosing when necessary with a different vector being delivered with different capsids on it?
Thanks, Cricket. So first, if I just take a step back, as many people have done over here, and just reframe the safety point around benefit risk. One of the experiences that I had just very recently prior to Affinia was being at Novartis Gene Therapy in Avexis, where we were able to bring Zolgensma to patients. And many of those children that were in my conference room at the time, if you look at the natural history, shouldn't unfortunately even be around. And to see them talking about being a pilot or being a ballerina in that conference room and talking about career aspirations was just unimaginable. We are now at the cusp where at least AAV9 in that one disease, spinal muscular atrophy, has shown promise. Of course, there are limitations, as with any new technology that emerges. In our case, we were cognizant that with conventional capsids such as AAV9, great as it is for spinal cord diseases such as spinal muscular atrophy, perhaps might not be sufficient to go into additional tissues, including CNS as well. The capsid work that we've done the most to date has been in-licensed from actually one of the co-founders of the company, Dr. Luke Vandenberg. He co-invented AAV9, developed much of the original technology at Mass Sinai. We've advanced it significantly since then. But what we've shown is actually with capsid manipulation engineering, we can actually get a few huge advantages. These, as Zandy also mentioned earlier, are 2 or 3 orders of magnitude differences in getting to different tissues, both RNA expression, in some case DNA as well. In our case, we use 2 very logically designed engineering methods. One is a method called ancestral sequence reconstruction, where you look at the phylogenetic tree of AAVs and essentially reverse engineer what the putative ancestors might be. This delivers structure-activity relationships that you can import into other scaffolds of AAV. And then secondly is a peptide insertion. Here, typically a 5 to 7 or a peptide, perhaps as big as 9 MERs, when inserted on some of the safe harbor anchor sites within the AAV capsid, give you a much bigger amplitude as well in terms of tissue tropism as well. So those are the items in which we're working on. I mentioned earlier, we also do work on promoter to get additional tissue and cell type specificity, as well as manufacturing, because we believe these three pillars side by side are essential for getting safety, efficacy, and also cost economics of the technology. On the question of redosing, this is a bit more complicated. Certainly, re-engineering capsid surfaces would hypothetically give you the ability to redose. But at least in our perspective, we think a more pragmatic and nearer-term approach might be the use of different pharmacologic agents prior to dose number 1, where you could blunt the initial cascade of events that happens to generate neutralizing antibodies as well as complement activation, and then potentially use the same or a second regimen that would allow dose number 2 to be administered. But I would say, practically speaking, redosing is a bit further away. I think we have some work to do and are seeing great successes in the ability to engineer capsids that can detarget the liver, alleviate that big liability, for example, of AAVs, and go much more into muscle and CNS as just classic examples in the near term.
Great. So I'm curious for all of our panelists, these outcomes, these adverse outcomes, are often seen really in the first in man or second and third in woman studies, and yet our preclinical models don't always predict what's going to happen. So I'm real curious, what are you doing to get us a better preclinical model, or is there no hope? Is it just a first in man and see what happens? Rick, you're smiling, so please give us some insights.
I wish I had more comforting news, but I think our preclinical models just aren't that great.
Yeah.
I think it is about continuing to let the experts work on that, but also to be very, very careful about trial design,
Yeah.
so that we minimize exposure to patients when we get to the clinic and learn.
Yeah.
Yeah, I don't think I have super encouraging news, at least not from within the walls of Bellicum today.
Okay, so with that depressive news, how do we pick the right time to give these potent therapies to patients who need them and not do it prematurely? In a cancer field, it might be a bit easier.
Yeah, certainly. Like most people, we're starting in very late stage disease with people with no approved options, and proceeding slowly and cautiously. Our HER2 program, for example, is enrolling patients who've exhausted all of the gamut of approved HER2-directed therapies and other therapies for their respective tumor types. And we're in our early dose escalation, enrolling patients sequentially with a waiting period after each patient to ensure that we're not seeing treatment-emergent toxicities given the HER2 expression in cardiomyocytes and pulmonary tissues. So far, so good. The safety profile has been great so far, but I will say it's a slow trial. So from a business perspective and for investors who are looking for meaningful data coming out of that trial, it's a little slow. But obviously our first priority comes to the
Safety.
patient and ensuring safety, and so that's how we've approached it.
Okay. What about gene therapy? Say, go ahead.
So actually, for in vivo liver gene editing, the non-human primate has actually emerged as a very good model. For lipid nanoparticle delivery, which is what we prioritize. When we started, we had a choice between viral delivery versus lipid nanoparticle delivery for the gene editors to the liver, and we prioritized the lipid nanoparticle delivery because we thought ultimately LNP could scale to the millions of patients we ultimately want to reach, where I think virus a number of different challenges. And so, so for LNP though, the mouse has not been a great model.
Right.
Things work in mice and have not worked in people, and vice versa. So, but non-human primates have actually emerged as a very good model. So all of our work over the last 3 years, getting our product ready to take to patients has been done largely in non-human primates, about 25 different non-human primate studies. And the lipid nanoparticle, the major issue is just when you infuse, there's an infusion reaction, an inflammatory reaction that can happen in a fraction of patients. But other than that, it's actually remarkably well tolerated, and it can be redosed. So it looks like, at least in this case, the NHP can be quite good. And I think Intellia Therapeutics has kind of paved the way here and shown that what works in non-human primates can actually translate well into human when they did their phase 1 study last year and had really remarkable results in terms of in vivo liver gene editing.
Rick and Sandy, please weigh in.
Yeah, so I would say a few things. Firstly, for capsids, we basically ignore mouse. And when we're looking at new capsids, whether systemic or CNS, we really look in NHPs. While that doesn't exactly tell you what's going to happen in humans, it's more indicative, and we don't have the models that are really available to look at efficacy, but we can at least look at the parameters that we want to be the case in humans in NHPs. So that's how we approach capsids. With promoters and regulatory elements and designing our vectors, we actually use organoids. So we have retinal organoids, we have motor neuron muscle organoids, developing CNS organoids, and that allows us to be sure that what we're expressing doesn't just express in the right cells in a mouse, that in the subtypes that we want it active in, we can see it active just in those cell types in human organoids, and it allows us to look on a cell sorting basis to make sure that what we're seeing is really appropriate for human cells. And that's the development aspect. But from the sort of strategy aspect, our programs at the moment tend to be local delivery. And in those cases, we use, in many cases, capsids that don't allow spread. It's the opposite of what people are searching for, capsids that go everywhere and deliver everywhere. So some of our indications are really local delivery, and you deliver just to the place you need it and avoid some of these potentially systemic effects. And in every one of those cases, you still need to be really cognizant of the potential safety issues. So we, over many of our studies, we've developed co-administration with different steroids that is very effective in allowing safe transduction and ultimately durable expression and efficacy.
Rick, any thoughts at the end there?
Sure. I think I'll support the previous comments made about, at least for AAV pharmacology, the best model that we have seen is non-human primates. Having said that, many of the disease models that the agency requires and that are available are typically mouse models. So when we do our screens as well as validation studies, we will typically start with a mouse model and then progress on to non-human primates. We're looking for cross-species translatability. It's indicative of human pharmacology as well for AAV. And we do need to step through the mouse model typically for the disease pharmacology and toxicology as well. In studies that one of our other co-founders has done, Botan Droska in Switzerland, he has shown very impressive data going from mouse to NHP to humans in the retina, preserved retina tissue. That the translatability from non-human primates for AAV in this market was more for promoters, but is actually very good going into humans.
So in academic institutions and laboratories, one of the approaches to look for off-target effects is not to scan the whole organ, but to do single cell sequencing to see if we've actually introduced a mutation that was unintended. That's a bit tough to do in patients. And so I'm wondering what you see as the gold standard for safety and efficacy. And I'd also like your comments on the recent guidance from the FDA in terms of what should be monitored even longer term.
Yeah, so the great news was about a month ago, the FDA put out guidance on human genome editing and really laid out their expectations for both the preclinical and the clinical programs. And I'll focus on their thoughts on the preclinical, and we've kind of heard this from a couple of different interactions we've had with them over the last 2 years. And our off-target editing analysis scheme has been developed based on this guidance. And so there are actually like 7 elements, and I wrote them down. And so the first is looking at multiple orthogonal techniques to get to a list of potential off-target sites beyond the on-target that you're intending to change. And then second, checking whether there is such off-target editing in primary human cells treated with drug at any of those potential off-target sites that have been identified. So in our case, for example, there are about 3,000 sites that we identified as potential off-target sites. Then we checked in primary human liver cells treated with drug and found no evidence for off-target editing at any of those 3,000 sites. And then you're supposed to check not just in the target tissue, but in other tissues where the drug might go to with in vivo systemic delivery. So where's the biodistribution of the drug, and check in those other cell types. So in our case, the drug mostly goes to liver, but then it also goes a little bit to spleen and adrenal cells. So we've checked in primary human adrenal cells, primary human splenic cells, and find very similar data in those cell types. In addition, you're supposed to check multiple lots of humans, not just donor cells from one lot, but multiple lots. So we've done that for multiple liver lots as well. In addition, you're supposed to check for germline editing. So are you getting editing in sperm or ovary cells, and to limit the possibility of germline transmission ultimately. And so we've done that. We extract the sperm from monkeys pre and post treatment and find no evidence of editing in the sperm. In addition to these analyses, we're supposed to check for structural variants, so not just single base pair changes, but much larger changes like translocations, and we find no evidence for that. And then you're also supposed to check batch-to-batch variability. So you could have differences in impurities leading to maybe differences in off-target effects by batch. So we've checked in process development batch and engineering batch and our clinical batch and find very similar, basically no observed off-target editing at any of those batches. And then lastly is, I think what Cricket was getting at, all of that work I just described is what's called based on guide-dependent off-target. So that means it's based on the guide sequence, or you're looking to see if there's any off-target. There could be guide-independent off-target as well. And what we've done to evaluate for that is do 500x whole genome sequencing in liver cells pre and post-treatment and find no evidence for global changes in terms of guide-independent off-target in this analysis. So it's a pretty long list, about 3 years of work, but this is really the bar that's been set by the FDA.
Mm-hmm. Rick Fair, I'm curious, when you think about off-target effects of cancers, perhaps the most important adverse issue would be that you don't kill the tumor. But as you pick more and more specific targets to direct these therapies to, you often find low-level expression in other normal and healthy cells. As a cardiologist, I'll tell you the heart is often the target of those unintended effects. So what level of expression? Is it the level of expression that is going to help make specificity of these cell therapies, or is there something more that we can do to improve safety of them?
Sure. So I can tell you, I'll use the HER2 example that I gave earlier in terms of what we did. I think we have multiple layers to try and get at this problem. One is about the selection of the binder. So making sure that we have a binder that isn't so high affinity that it will find even low levels of expression of HER2. And differential expression sort of defines a good target. And so high levels of expression of HER2 in tumor, lower levels in cardiomyocytes and elsewhere with a moderate affinity binder, we think gives us some safety margin. But that's probably not enough. So that's where we added our molecular switch controllable technology, both controlling activation of cells, and if we start to see low-level adverse events that are concerning, we can withhold the activating agent. The cells become quiescent, and we potentially can return the patient to treatment subsequently. Or if we have a high-grade adverse event where we believe that the etiology is about CAR-T attacking healthy tissue, we can eliminate the cells via our safety switch via apoptosis. So I think that's the approach we've taken. Others are looking at logic gating, so there's no or very few highly selective tumor antigens. So, but there are potentially and/or logic that one can use to say, if I see this antigen and I don't see this one, then I'm not at the heart, I'm at the tumor. And while those approaches are novel and really only entering the clinic now, I think there's a lot of optimism that that might be helpful as well. But I think multiple ways of attacking it, but it's clearly an issue that's a priority for us and others.
So one of the issues that in the cardiovascular arena, we've recognized that the consequences of even a subtle change are not manifest for years and sometimes decades, and then the cardiomyopathy and heart failure ensues. So how long do we need to monitor these therapies? And I'd be curious from each of you what you predict will be the long-term sequelae assessment.
Well, as you know, based on FDA guidance, we're monitoring patients for 15 years.
Yeah.
We're obviously a young company, so we don't have long-term data yet, but I would say that's a good starting point, and we'll see what we see in that time frame.
The FDA guidance for gene therapy has been 15 years, and then the most recent guidance for human genome editing was also 15 years.
Yep, same.
Similarly, from at least the Zolgensma experience, the longer-term registries are instrumental. Although I would not discount some of the long-standing preclinical work, both histopath as well as letting those studies also go as well to look for any potential durability effects or side effects.
So I guess related question would be, what is that going to do with patient compliance and uptake of these life-saving therapies and cost of delivering them to the healthcare, from the healthcare system?
Well, I'll say in the CAR T setting, to date, at least in the solid tumor setting, we haven't cured patients with CAR T cells, and so these patients are being seen in the long term by their oncologists. Hopefully we see really long survivals and they keep coming back to their oncologists and get a clean bill of health, but it does allow us to monitor these patients. I would say the long-term follow-up in our studies doesn't add a tremendous amount of cost to the cost of studies, and I wouldn't expect that to be the case in the commercial setting. So I think in our area of the world, I don't think that those are real big drivers of concern.
Great. Sandy?
I agree. I think when you've shown good efficacy and safety acutely, and you have registries to follow up in the long term, it's really that benefit in the context of safety which drives physicians and patients to use your product. And with respect to cost, I think the cost for these products manufacturing, bringing that down over time is really important, and the size of the populations that you're treating.
Yeah, I think there's a general discussion about for a therapy that's one and done, lifetime of benefit, one-time treatment, how should they be priced? And I think typically when people think about these therapies, gene editing or gene therapy, they think about rare disease pricing, so millions of dollars a dose. I think, for us, for cardiovascular disease, which is where we're hoping to, really have an impact, our pricing won't be that. It won't be millions of dollars a dose because we're trying to reach millions of patients. And so the question is, what is it? And that, will depend a lot on cost of goods. And the good news there is that our drug looks very much like a COVID vaccine. So it's an mRNA packaged in a lipid nanoparticle. And we know that COVID vaccines can be made for a couple of dollars a dose. So this is a very fortuitous thing, actually, in terms of our manufacturing costs over the last couple of years, because when we started in 2018 with this concept of mRNA packaged lipid nanoparticle, we actually didn't know how low we could get cost of goods to. But COVID has really changed all that and made it very line of sight to very manageable cost of goods. So I think we'll be able to ultimately price our product in a way that can be accessed by lots of patients all over the world.
Rick?
Important question. Both the societal affordability part of it, as well as the health economic argument behind it. The reason we're focusing on capsids, promoters, and manufacturing is to be able to find these capsids that are super effective, meaning you can use a much smaller dose to start with, which will make a big difference. Similarly on the promoters. And then lastly, also in terms of manufacturing technologies and processes, we do believe there's ample room for improvement there, potentially to get some of these costs for gene therapies, AAV delivered, to be in the same realm as biologics over time. But very important question, and one that I think us as sponsors, as well as the system at large still needs to solve.
Fantastic. Well, we're almost at time, and I want to thank our terrific panelists for joining us here today. Surprisingly, we had no questions, so I'm sure you're going to see all of our panelists out in the auditorium to speak with them. The next session that will be up will be RNA technologies. What have we learned, and what are the lessons that we take forward from that? And that will be spearheaded by Janet Wu. Thank you very much.
Thank you.